Hello students, today we will be talking on a very important topic called general properties of carbohydrates. Carbohydrates are the single most abundant class of organic molecules found in nature. They act as storehouse of chemical energy. They also act as component of supportive structure in plants, crustacean cells, and other connective tissues in animals. They are the metabolic precursors of virtually all other biomolecules. Conjugates of carbohydrates with proteins and lipids perform a variety of functions including recognition events that are important cell growth, transformation and other processes. Not only that, they are also essential components of nucleic acid such as D-ribose and 2-deoxy-D-ribose. So in this session today, let us discuss about some biological properties governed by the carbohydrates along with its medical implications. So general properties of carbohydrates. Carbohydrates are versatile class of molecules. When the word carbohydrate was coined, it was referred to hydrates of carbon, that is compounds of the general formula CnH2ON. Chemically, carbohydrates are defined as polyhydroxyaldehydes or ketones, or substances that yield such compounds on hydrolysis. Carbohydrates play a number of important roles in biochemistry. First, they are major energy sources. Second, oligosaccharides play a key role in processes that take place on the surface of the cells, particularly in cell-to-cell interactions and immune recognition. So now, physical properties of carbohydrate. Carbohydrates can be easily divided into four main groups called monosaccharide, disaccharide, oligosaccharide, and polysaccharides. Some of the general chemical properties of carbohydrates are discussed herewith under different subheadings as follows. Number 1. Oxidation reaction of carbohydrates. Oxidation and reduction reactions of sugar play a key role in biochemistry. Oxidation of sugars provides energy for organisms to carry out their lab processes. The highest yield of energy from carbohydrates occur when sugars are completely oxidized to carbon dioxide and water in aerobic processes. The reverse of complete oxidation of sugar is the reduction of carbon dioxide and water to form sugar by a process called photosynthesis. Several oxidation reactions of sugar are important in laboratory practice because they can be used to identify sugar. Aldehyde groups can be oxidized to give the carboxyl group that is characteristic of acid, and this reaction is the basis of a test for the presence of aldoses. When the aldehyde is oxidized, some oxidizing agent must be reduced. Two types of reagents are used in the laboratory to detect the presence of reducing sugar, which are based on oxidation of monosaccharides. The first of these is Tollens reagent, and the second one by using an enzyme called glucose oxidase. In the Tollens oxidation of glucose, alpha-D-glucose is oxidized to a compound called lactone using silver ammonia complex ion as an oxidizing agent. Here, reduction of silver ion to silver metal and deposition of free silver as a silver mirror indicates that the reaction is taken place. A more recent method for detection of glucose, but not for other reducing sugar, is based on the use of enzyme called glucose oxidase, which is specific for glucose. So next, esterification reaction of carbohydrate, that is formation of phosphoric ester. Phosphate esters are frequently formed by transfer of a phosphate group from ATP, that is adenosine triphosphate, to give the phosphorylated sugar, and ADP, that is adenosine diphosphate. Such reactions play an important role in the metabolism of sugar. Molecules showing mono and diphosphoric ester, which are important intermediates in the metabolism of monosaccharides, are also shown here. Formation of glycoside. It is possible for a sugar hydroxyl group, that is ROH, bonded to the anomeric carbon to react with another hydroxyl group to form a glycosidic linkage. This type of reactions involve the anomeric carbon of the sugar in its cyclic form. The peranose and furanose form of monosaccharide react with alcohol in this way to form glycoside. The new bond formed between the anomeric carbon atom and the oxygen atom of the alcohol is called glycosidic bond. Glycosides are named according to the parent monosaccharides. Glycosidic bonds between monosaccharide units are the basis for the formation of oligosaccharides and polysaccharides. Glycosidic linkage can take various forms. The anomeric carbon of one sugar can be bonded to any one of the hydroxyl group on a second sugar to form an alpha or beta glycosidic linkage. Two ways in which two alpha-D-glucose molecules can be linked together are alpha-1,4 and alpha-1,6. The difference between esters and cellulose depends on the glycosidic bond formed between the glucose monomers. Glycosidic bonds showing alpha-1,4 and alpha-1,6 linkage. Here, glycosidic bonds showing beta-beta-1,1 linkage. Glycosidic bonds showing alpha-1,2 linkage in the case of sucrose. This is the structure showing lactose having glycosidic bonds between beta-1 and 4. Glycosidic bonds showing alpha-1,4 linkage in the case of maltose. Carbohydrate can also be converted to several derivative forms. Here, a variety of chemicals and enzymatic reaction produces derivatives of the simple sugars. Some of the most common derivatives of carbohydrates are discussed here. Number 1, amino sugars. Amino sugars are an interesting class of compounds related to monosaccharides. Amino sugars contain an amino NH group in place of hydroxyl group. Only 3 amino sugars are common in nature. They are D-glucosamine, D-manosamine, and D-galactosamine. N-acetyl D-glucosamine, a derivative of D-glucosamine, is a component of many polysaccharides, including connective tissues such as cartilages. Number 2, oxidation to aldonic acid, also otherwise known as sugar acids. Aldehydes are oxidized to carboxylic acid by several agents, including oxygen. Similarly, under basic conditions, the aldehyde groups of an aldose can be oxidized to a carboxylate group. D-glucose, for example, is oxidized to D-gluconate, the anionic form of D-gluconic acid. Any carbohydrate that reacts with an oxidizing agent to form an aldonic acid is classified as a reducing sugar. It reduces the oxidizing agent. By quantifying the amount of oxidizing agent reduced by the sugar solution, one can accurately determine the concentration of the sugar. Diabetes mellitus is a condition that causes high level of glucose in urine and blood, and frequent analysis of reducing sugar in diabetic patients is an important part of the diagnosis and treatment of this disease. So next, oxidation of uronic acid. Monosaccharide can also be oxidized enzymatically at carbon number 6, yielding uronic acid, such as D-glucuronic acid and L-iduronic acids. For example, enzyme catalyzed oxidation of D-glucose yield D-glucuronic acid. Here shows the reaction in both its open chain and cyclic hemiacetal forms. The intravenous anesthetic propofol, for example, is converted to the following glucuronide and then excreted out of the body through urine. So next, reduction of carbohydrates to alditols. Carbonyl group of a monosaccharide can be reduced to a hydroxyl group by a variety of reducing agents, including hydrogen in the presence of a transition methyl catalyst and sodium borohydride. The reduction products are known as alditols. In this way, a sugar alcohol is formed when a carbonyl group is reduced to a hydroxyl group. Reduction of D-glucose gives D-glucitol, more commonly known as D-sorbitol. Here, D-glucose is shown in the open chain form. Sugar alcohols can be useful as a dietary substitute for a lot of reasons. They have less calorie content than ordinary sugar and cannot be digested by bacteria present in the mouth. As a result, candy that is sweetened with a sugar alcohol is less likely to contribute to tooth decay. For this reason, many products such as chewing gum are sweetened with sugar alcohol. Sorbitol buildup in the eyes of diabetic person is implication of cataract formation. So next is deoxy sugars. The deoxy sugars are monosaccharides with one or more hydroxyl group replaced by hydrogens. 2-deoxy-D-ribose, whose systematic name is 2-deoxy-D-erythropentose, is a constituent of DNA in all living things. Deoxy sugars also occur frequently in glycoproteins and polysaccharides, L-fucose and L-rhamnose. Both 6-deoxy sugars are components of some cell walls, and rhamnose is a component of uaven, a highly toxic cardiac glycoside found in the bark and root of uabio tree. Uaven is used by the East African Somalis as an arrow poison. However, the sugar moiety is not the toxic part of the molecule. Acidic polysaccharides are a group of polysaccharides that contain carboxyl groups or sulfonic ester groups. Some examples of acidic polysaccharides are summarized herewith. A. Hyaluronic acid. Hyaluronic acid is the simplest acidic polysaccharide present in connective tissues. Hyaluronic acid is composed of D-glucuronic acid joined by a beta-1,3-glycosidic bond to N-acetyl-D-glucosamine, which is in turn linked to D-glucuronic acid by a beta-1,4-glycosidic bond. B. Heparin. Heparin is a heterogeneous mixture of variable sulfonated polysaccharide chains ranging in molecular weights from 6,000 to 30,000 grams per mole. Heparin has many biological functions, the best known and fully understood of which is the anticoagulant activity. It binds strongly to antithrombin 3, a plasma protein involved in terminating the clotting process. A repeating pentasaccharide unit of heparin is shown here. So now let us shift our discussion to some other interesting topics of carbohydrate properties. That is A. Testing of glucose. In type 1 diabetes, the blood does not produce enough insulin, whereas in type 2 diabetes, the issue is not lack of insulin but high blood concentration of sugar, that is hyperglycemia caused by insulin resistance. The analytical procedure most often performed in the clinical chemistry laboratory is to determination of glucose in blood, urine, or other biological fluids. A rapid test for blood glucose level is critical for early diagnosis and effective management of this disease. In addition to giving result rapidly, a test must be specific for D-glucose. That is, it must give a positive test for glucose, but not react with any other substances normally present in biological fluids under test. Today, blood glucose level are measured by an enzyme-based procedure using the enzyme called glucose oxidase. This enzyme catalyzes the oxidation of beta-D-glucose to D-gluconic acid. B. Honey. Honey is the first sweet known to humankind, and it is the only sweetening agent that can be stored and used exactly as produced in nature. Bees process the nectar of flowers so that their final product is able to survive long-term storage at ambient temperature. C. Galactosemia. Out of every 18,000 infants, one is born with a genetic defect that renders the cell unable to utilize the monosaccharide galactose. Galactose is a part of lactose, that is milk sugar. When the body cannot absorb galactose, it accumulates in the blood and in the urine. This buildup in the blood is harmful because it can lead to mental retardation, failure to grow, cataract formation in the eyes, and in severe cases, death due to liver damage. When galactose accumulation results from a transient disorder in infant, it is known as galactosuria. It has only mild symptoms, but the enzyme galactose 1-phosphate uridyl transferase is defective. The disorder is called galactosemia, and its symptoms are severe. So in conclusion, derivatives of triose, tetrose, and pentose and of as 7-carbon sugar pseudoheptulose are formed as important metabolic intermediates in glycolysis and the pentose phosphate pathway. Pentose are important in nucleotides, nucleic acid, and several coenzymes. Glucose, galactose, fructose, and mannose are physiologically the most important hexose. And among the polysaccharides, the structural polysaccharide is cellulose, while functional polysaccharides include stars in plants and glycogen in animals. So here comes the end of this session.